Yes. Okay. Good morning, everybody, once more. I hope you had a pleasant night. And now I'm officially opening our stakeholder session. And thanks for the online participants to join us as well. Sorry. If you haven't met me yet, I'm Çağla from European Forum of Farm Animal Breeders, and I co-lead this communication dissemination and stakeholder engagement together with my colleagues at EAP. And we will try our best to have a nice and fruitful meeting. So what we want to do is to meet our stakeholders, because now we are nearly at the end of our project. Yes, this is. And we want to share some results that we have, and we would like to get your feedback on how we can facilitate more the uptake of our results for the benefit of the sector overall. And we have a full agenda. So we will have a summary of our project presented by our coordinator, Elisabetta Giuffra. And then we would like all the stakeholder participants to introduce themselves very basically, not like a presentation, but who you are, where are you coming from, which organization. And then we will have a presentation from Peter from PIC about overview of the functional research areas of PIC. And then we will have a discussion part where we can ask each other questions in order to facilitate or help us, support us in disseminating our results much better and much more efficient to the sector. And then we will have a networking coffee. So before giving the floor to Elisabetta, I would like to share first our video that we prepared for Eurofang from the Eurofang cluster of 6 projects. Can you play it, my friends?
Eurofang aims to address challenges around farmed animals by unraveling the connection between the genetic makeup of animals and their traits. Across different climates and agrosystems, animals display different physical characteristics, allowing them to be adapted to different environmental challenges. Variability in DNA is one of the things that causes this variation, together with the environment. The complete set of DNA of an organism, the genome, is made up of approximately 5% genes and 95% intergenic, or so-called non-coding regions. These intergenic regions, including regulatory elements, modify how and when a gene is switched on, which can strongly influence traits that are important for welfare and production. We know that the regulatory elements play an important role, but we are not sure in what way, and how they are in turn influenced by epigenetic changes coming from, for example, the environment or food intake. This is why developing an encyclopedia for this 95% of the genome is so important. This encyclopedia is called Functional Annotation. Functional annotation allows scientists to map the coding and non-coding features of the genome. With this information, animal breeders can select and breed animals that can cope better with a changing environment and meet societal expectations. The Eurofang initiative is part of the Worldwide FANG Consortium and was initiated by 3 Horizon 2020 funded projects. Eurofang continues to grow and now comprises more EU-funded projects. For more information, visit the project websites and follow the Eurofang social media.
As well. So, and yeah, we would appreciate if you can also share it with your network, with your clients. It's really, it makes it easier to understand for people not coming from the research like me. So now I give the floor to, I just want to remind one thing, that we will have a final meeting, probably 7-8 November, probably in Brussels. So, but we will soon share with you, save the date. So, but mark your agendas already. So, Elisabetta?
Thanks everybody for joining this event. And I will try to give some, in a brief time, in fact, so it will be maybe a bit too much synthetic, some flash information about this project. And the main aim of this project that started in July 2019 and will end at the end of this year has been to deliver underpinning knowledge on the pig and chicken genomes, and also to make work to enable the translation of this knowledge for the benefit of the pig and poultry sectors. And we are a consortium that is not so big for a European project, but quite complete and diverse. As you can see on the left, we are composed of research institutions, but also the biotech, small medium enterprise, large breeding companies. We have breeder associations, international government organization, a project engineering and technology transfer company. And you can see all the institutions, all the partners that are listed on the right. And in the course of the project, we are also intensively, collaborating with 3 breeding companies and associations that are Aviagen, HyperBV, and IFIP. And GENE-SWitCH has been built around 3 specific and interconnected aims that are to produce more functional annotations across tissues and developmental stages. This is really the landmark of this project, compared also to other projects that are made on different species in Eurofund, which is the work of the first 3 work packages of the project. Aim B is using these annotations for precision animal breeding, where we took 2 very different approaches that I'm describing briefly now. And C, to work on the standardization of data and processes, dissemination and outreach. And the underlying questions behind these aims have been, can we identify and characterize the functional genomic elements, those that are switched on or off during development, and that contribute, we don't know to what extent, and we try to find out, to the phenotypes, also the adult animal, and what's the relative impact of their genetic variation on the relevant, of the main production traits that we could investigate. And you see also in the bottom right of the slide, the addresses where you can find more information, more detailed information. And I really recommend you to find online, and maybe we can provide the links on the screen at some point, the address of the video you just saw, because it's like, I mean, we really like it, as Chagla said, it's a good starting point for any user. And on AMA, as I said, it's the functional annotation across tissues and developmental stages. And here you see that we chose in both species equivalent point of development, reflecting early organogenesis in the embryo or fetus, late organogenesis, newborn or hatched. And then we had also in the database of FUNG that are in open share and open to anybody, additional data reflecting the adult stages. And functional annotation is exemplifying this scheme where you have a gene and you may want to know, in fact, the annotation of its promoters, the enhancers that regulate its expression, how many isoforms this gene is expressing, and so on. And the specific aims of this work package are exemplified here, to identify these elements, to characterize their dynamics that we call switches. So we are interested really to see which gene, for example, is activated here, but it will be switched off here. And this is a part of fundamental investigation of this project. So to really characterize the pattern of conservation and variation in mammals versus birds, etc. And to organize all this information to deliver high quality and richly annotated genomic maps that are visualized in the ensemble interface, where you have, for example, we will have by the end of the year or around the end of the year, the ensemble regulatory build for the pig and chicken genomes, including the broiler and white leghorn genome, that will be really one of the main outputs of this project. And here you see a typical representation that you can have of a genome when you focus on a region and you have all these annotations available. The example is for human, I think. And of course, these annotation maps and information are shared with the following work packages that realize aim B to use these annotations. And in this, we took, as I said, two different approaches. Sorry. One is the integration of genetic data on the variation that you can see by SNPs, for example, in this presentation. And it's focused on genomic selection. So on the question, can functional annotations enhance the prediction accuracy of breeding values in commercial populations? This has been realized in the project by a large work package, which is work package 4. And in the other approach, it was more exploratory and I'd say complementary. It's on the integration of epigenetic data and to see the effect of diet, which is a major environmental factor and very important in animal breeding, on the epigenetics of two main tissues, liver and muscle in the pig. And the idea behind, there is echo now? No. And the idea behind was to provide the basis for future studies focused on better farm management using lower quality, more sustainable feed as an example. And this is work package 5. On this work package 4 that I said, the idea was to improve predictive models for genomic selection. You see the partners who are in charge of this work package to the right, led by Hendrix Genetics and co-led by Wageningen University. And in this work package, they extended genomic prediction models to exploit new annotation maps of pig and chicken that were generated. And they used the development of new genomic prediction models, generation of fine-mapped QTL and DQTL, especially in the pig. This is published and is being published in the chicken, and validating the models in large-scale commercial data that is in progress and will be probably completed by the end of the project, so in time. In the diet for epigenetics, I will be a bit faster here, but we went to see the influence of maternal diet on the epigenetic programming to understand if different fiber contents in the diet had could really affect the epigenetic and transcriptional picture of porcine liver and muscle in both fetuses and piglets. And here I put some specific information, but I will go fast because otherwise I should talk too much in detail. And I invite you to go to see the more information we find online on this aspect too. And AIM-C is about standardization of data and processes, dissemination and outreach. So we work in FAIR, under the FAIR principle, findable, accessible, interoperable, reusable data and results. So it involves data coordination, curation, validation, archiving, dissemination, and publication through the European Biological Data Archives and the Farm Data Portal that provides single centralized access to all the information, and where we have a dedicated GENE-SWitCH page. There has been extensive dissemination, communication, and training activities to ensure mutual interactions with all stakeholders. This is one of those events we have tried to make. And since the beginning of this project, we are among the founders of these Eurofund initiatives, but now has been much developed. And since January, the Eurofund research infrastructure has started, which is a new project where several partners of the same project are participating. And then I prepared also, the impacts. There is a slide on impact, in fact. These are the impacts that we have promised. So deliver comprehensive genome annotation maps of high quality in the targeted farmed animal species. We want to pave a way for subsequent use of annotated genomes to improve precision breeding in farm and animal production. There are also other projects that are starting as a follow-up, but we use the heritage of GENE-SWitCH. We have provided progress in understanding genotype and environment interactions and deciphering the mechanism of how environmental and stressors could be transmitted across generation. We contribute to the international cooperation of genome annotation, which is the Global Fund initiative. And in the long term, we hope that we contribute to enhance the sustainability of farmed animal production. And I underline again that FANG has provided the opportunity to increase these impacts in Europe and beyond. And I think I will stop here, because here I also put a kind of synthesis of our results that, however, maybe we will use these slides if the discussion move on, because I prefer to be faster, because I had 15 minutes and I think I used them, correct? This is what we see as exploitable results of the project, for example, for fair resources and, well, and from the different work packages, but I think it would be too complicated to get into detail. So, Chagla, I think I stop here.
Can I invite the invited stakeholders here, so that, because we don't have a microphone that is portable, so you can introduce yourselves here shortly? Barbara, yeah, or you can have a seat. Barbara, Marie-Jose, Anna, Emanuela, Paul. And maybe we can also ask our online participants, Peter and Anna, to shortly also introduce themselves after the participants here. Yes, Barbara, please.
Yes, good morning. My name is Barbara Halitius. I work for Topics Northwind Research Center in the Netherlands. We are a pig breeders association, and I'm responsible for a platform as a platform leader of our genetics and genomics platform.
I go on, maybe?
Yes, please.
Okay.
Sorry.
So hello, I'm Marie-Josée Mercave from IFIP, the French Institute for Pig and Pork Production. So I'm working in close connection with the breeding companies member of Alliance R&D in France, the main French breeding companies. And also I'm involved in the preservation program of the local breeds.
Thank you.
Hi everybody, my name is Anna Concollato and I work as a senior policy officer for Italia, which is the main Italian organization for political and trade union representation for meat and egg agri-food chain, whose goal is to join forces to create a system proposing the integrated supply chain, in which all the company we represent, about 30, have the complete control of the production processes, and with a view to sustainability and transparency for consumer. The mission of Italia is to represent and protect associated companies towards institution and administration, but above all to enhance and promote their product. Italia represents the 90% of the national poultry meat production, 20% of egg and pig production, and 40% of rabbit production.
Thank you so much, and also for the information about Italia. Paul, well, everybody knows you, but...
Yes, I'm Paul Betcher from Food and Agriculture Organization of the United Nations, which is, I guess, better known as FAO. And at FAO, I work in the Animal Genetic Resources Unit, and basically help support countries. Let's say most of the support is to developing countries in managing better their livestock genetic diversity, to both conserve it as well as utilize it to improve food security and livelihoods.
Thank you, Paul. Emanuela?
Yes, good morning, everybody. I'm Emanuela Parlato, and I'm animal geneticist working for the PIC, Pig Improvement Company. My role in the PIC is working the genetic service team and supporting our customers in the success of their genetic program.
Thank you very much. So we can also have a short introduction. Anna, can we start with you?
Yes, sure. Which Anna? Me?
Yes,
Online?
you Anna, Anna Granado.
Because you are looking at the opposite direction. I mean,
No,
anyway,
screen is-
I understand you have the screen behind you. I'm sorry. I was taking some notes. So, yeah, hello. Good morning, everyone. So I'm one of the colleagues of Chala and Gina that are with you there in Rome. So I'm the director of IFAF, and so the European Forum of Farm Animal Breeders. We represent the interest of animal breeders here in Brussels. And we have also this technology platform, FarberTP, which is representing scientists so from both sides, academia and private sector here in Brussels too. Happy to be here today with you. Hello.
Thank you. Stefania?
Good morning, everybody. I'm Stefania Retano from the Ministry of Agriculture, Food Sovereignty and Forestry of Rome. I work in the office that follow and supports the animal breeding sector in Italy, both genetic improvement and protection of animal biodiversity. We work closely with all the Italian Breeder Association that manage herd books in Italy. And I thanks to be here with you today.
Thank you, Stefania. So the floor is yours, Peter. You can also introduce yourself.
As usual, hello, good morning. I'm Peter Knap. I'm a colleague of Emanuela working for PIC. I am the genetic strategy manager in PIC. And I'm going to do the entertainment for the next 15 minutes. So we'll try to share my screen and then we see where we go. All right. We talk about our experiences with functional annotation in pig breeding. And this talk is really due to my colleague Bruno Valence. Bruno and I, we are members of a team of 12 people who deal with genetic improvement in PIC. Most of them are based in Tennessee, in the United States. That includes Bruno. In fact, I'm the only member of the team who lives in Europe. And that is why I am giving this talk now, because Bruno is asleep in Tennessee. Now, when we talk about animal breeding programs, we should recognize that there are plenty of bits of functionality that have to be taken care of. And for the next 15 minutes, we focus on box 4, breeding value estimation, and a little bit on genetic models. That has a lot to do with working package 4 of the GENE-SWitCH project, of course. When we look at breeding value estimation in livestock and the history of it, we notice that over time, the technology that we use for breeding value estimation changes from technology that is biology driven to technology that is statistics driven, and then back again, etc., etc. Right now, in most of the animal breeding industry, we are straight in the middle of genomic prediction that was kind of started up something like 13 years ago. Now, 7 years ago, I did a lecture in the University of Göttingen in Germany. And I told these people that it is not very likely that a few hundred markers will capture much genetic variation in traits of interest in livestock breeding, which means that we need more of them than a few hundred. or they should not be random, or both, of course. And as an example, in those years, that was in 2015, I looked at the 1% of the 60,000 SNP markers that we were using at that point in time, the 1% markers that were most influential for a particular trait. And I argued in Göttingen that these will be different markers for different traits in different populations. So they are not random. And it looks like this. We have 7 big populations here, from left to right, A to J. And every data point represents a particular trait. They have 4 different colors because you can subdivide them into 4 different trait categories. And on the vertical axis, which is logarithmic, as you can see, we have the proportion of the genetic variance of each trait that could be ascribed to those 600 SNPs. We have a few cases there on the top of the graph where those, let us say, major markers explain a large part of the genetic variance. And that would, of course, lead to the option of giving those particular SNPs more focus in breeding value estimation, in genomic blood. That is the situation of, let us say, 7, 8 years ago. And since then, we have moved on. We are focusing more on biology now in what John Williams used to call quantomic prediction. And that has a lot to do with moving away from the black box of genomic prediction as we are doing it now, where we don't really know what the SNPs do. And in fact, we are not really interested in that because the main thing we want to do is making it work. And it certainly works. But the next step in the whole technology development will be the inclusion of biological knowledge. Now, research and development in a breeding company must, of course, bring value to the breeding program. And the R&D that we do in PIC takes place in terms of the boxes that I've colored red here. Certainly not only breeding value estimation. I myself spent most of my time on in box 2, breeding goals, and where are we taking the populations to. And there are plenty of other things with phenotyping in box 3, things to do with avoidance of inbreeding in box 5, and managing genetic diversity in box 7. But here we talk about breeding value estimation. And then there are multiple fields here. Quantitative genetics is classically the thing to use, and genomics is becoming more and more important, as we all know, often by exploring genomic annotation. And for that, we do our research internally, and we have external collaborations with academia. The internal research typically focuses on one or just a few genes with a known role and a known relationship with a trait of interest, or suspected relationship with a trait of interest. Suspected, for example, because of previous human studies or knockout experiments, typically in mice. And we do a lot of GWAS analysis to establish, using CBI and the ensemble databases, to establish the things that we want to know. That leads to multiple population sequence data to find variants in the gene that are segregating in our populations, to evaluate those variants in terms of functional relevance. For example, in terms of location, where they are, what type of variant they are. If it is a SNP and an exon, is it non-synonymous? If it is an indel and an exon, is it frame shifting? And if so, how large? We work with shift scores, etc. All the typical stuff that you have been working with for a long time now. To highlight the most relevant variants, to verify patterns of segregation across populations, and answer the question, always very important, are the alleles that we focus on, are they indeed more frequent in the populations where we expect them to be more frequent? Nothing new here. When we talk about collaboration, I'll give you a few examples. We have a long-standing relationship with the Roslin Institute in Edinburgh. And when John Hickey worked there, we started on large-scale sequencing across 9 of our breeding lines. That started late 2015. Direct sequencing of subpopulations at varying depth, imputing the variants to the full populations, and explore those sequence variants to see if they lead to better prediction of the additive genetic value of our animals for the breeding value estimation. That was based on statistical information, not an annotation. We just looked at the top SNPs from previous GWAS exercises, and of course we studied linkage disequilibrium. That project came to an end something like 2 or 3 years ago, and we are still working on it, Bruno and his colleagues, to explore the whole thing further, this time using annotation. 3 of John Hickey's colleagues have been working on that data. Sequence peaks of 9 populations, and they have been looking at private variants, that are variants that segregate in one of the 9 populations only. And on the other extreme, widespread variants that segregate in all 9 populations. And of course, there is a lot in between. And they looked at associations of the genotypes with 3 simple production traits like growth rate and body composition. And they found that the private variants, that is in the bar chart, the variant with prevalence level 1, so they are segregating in one line only, they are more often functionally relevant than the widespread variants. What you see is that the blue bars are getting lower when we move to the right-hand side of the graph to more widespread variants. But they show much lower minor allele frequencies than the widespread variants, as you see on the graph on the right-hand side. In the various types of variants, the classification of the variants, we see in practically every category that the MAF is much larger in variants that are not private, which means if you multiply that together, that those private variants contribute much less to the phenotypic variants for most of the stated functions. And we see that in this table here. Again, the private variants are on the left-hand side, and we move to more widespread on the right. And the darker colors, the cells with the darker colors, they indicate that a larger proportion of the phenotypic variants is explained here. So in this paper, we concluded that accounting for private variants and other low prevalence variants is unlikely to benefit a crossbreed analysis such as the prediction of genomic breeding values in a population if you're using reference populations of a different genetic background. And that was very important for us to establish and to quantify, because we do breeding value estimation that way. So that was an important thing to get out of this exercise. But as you all have noticed, it has nothing to do with annotation. Up to now, PIC has not been using annotation for breeding value estimation. And that is going to change. We just started another collaboration with academia, a project titled Genomic Evaluation Using Functional Genomics Data at Individual and SNP levels. And the collaborators are Hao Cheng of the University of California, Dani Lorenzo of the University of Georgia, and Andres Lagarra, who works in industry, who started working in industry recently. So all that is going to happen in America. When Elisabetta told us about GENE-SWitCH 20 minutes ago, I recognized a lot of functional overlap there. We aim to explore the sequence data with Fang-enabled annotation, categorical annotation, and continuous annotation. We will look at weighting the SNPs in breeding value estimation derived from the annotation data. Of course, statistical models will have to be developed and implemented and evaluated with Dani's BloodBirth 90 software and the JROS that Hao Cheng has been working on. And that means that here we plan to go and use annotation in breeding value estimation with differential weighting of SNPs in a new version of genomic blob. And that comes back a little bit later. These same three guys have been working on a very focused bit of sequencing in the CD163 gene of the pig. CD163 is the protein in the pig that is hijacked by the PRRS virus for making the virus, for enabling the virus to reproduce itself. Without that pig protein, the PRRS disease cannot exist. So it's an interesting thing from a sustainability point of view and a disease resistance point of view. We have been looking at pooled sequencing of the exons in that gene and scanned them for naturally segregating variants that would damage the protein function. Because if the protein function is damaged, then the pig would become resistant to the virus in particular ways. Variants like stop gain mutations or frame shift indels, so the really heavy the really heavy variants. And we could not find such variants. In our lines, the CD163 gene is practically non-polymorphic. So instead of exploiting those variants that do not exist, one of the alternatives would be to make them. And then, of course, that leads us to genome editing. In our internal research, we do quite a lot of scanning for those naturally segregating candidate variants, not only for the CD163, but any kind of gene of interest using historic sequences, highlight strongly relevant variants, large exon deletions, for example, or non-synonymous SNPs and highly conserved areas, and access the segregation in our current populations, identifying live animals carrying the relevant alleles, and then trying to take it further. And that is 18 minutes, not bad. And so we give it back to Celia, and we can come back to all this in the discussion that will follow now. Thank you.
Oh, thank you so much, Peter. It was interesting to see how PIC is using or starting to use the functional research. Yeah, we are a little bit before our agenda, so it's good. Now I would like to invite Andrea here. And I would also like to invite our invited stakeholders here so that it would be easier for you to ask. And yes, sorry about the logistics. We want you to make the morning exercise. So already some questions. So, oh, sorry.
Can you use mine? Oh, this is what is going there? No. Okay. Can you use mine? Do you mind?
Yeah, it was just for me to have some questions on hand.
Jullie had nooit snakken heen. Ik forstår, hvad det var. Det er bare sådan,
Is there any issue? I don't know. Anyway, good morning. My name is Andrea Rossati. Thank you, Chagla, for presenting me. I'm the Secretary General of EAP, the European Federation of Animal Science. I see some friendly face in the room and also here on the table. And I was asked here to moderate the discussion. And in few words, I'm the obstacle for you between now and the coffee break. So we may try to make it a little alive this time that we have, and using all the possible time to discuss about the outcomes and the research done by this project, GENE-SWitCH, and the policymakers and representatives as stakeholders that we have here today. They were all presented before. Hi, Peter. Good to see you even online. I was hoping you were coming into Rome, but maybe for next time. Peter, are you online?
Next time will be fine. Yes, I'm online, but I got muted.
Okay. Okay, good. So I will start making some questions, but I will make a first question that come out of the Peter presentation, and that was just an idea I got 2 minutes ago. I mean, a question coming 2 minutes ago, but I will also expect that you also make questions and you also answer and make questions. So it should be quite kind of a live discussion. And Peter ended his presentation talking about genome editing and just very general discussion. How can you think that the genome editing can be supported by the results or the research made by GENE-SWitCH? Very general discussion. Does anyone want to answer? Yeah, the online people. I mean, Peter and Anna or other people there.
I can try to kick it off if you want.
Please, Peter. Yes.
One of the things that gene editing has been a hot item in the pig sector in terms of two important diseases. You can make a pig resistant to PRRS by gene editing, and the idea was that it would be possible to make a pig tolerant to African swine fever by gene editing. The second part was researched in the Roslin Institute again. That did not become a big success. The edited pigs, they were not tolerant to the disease. But in both cases, the focus was on a particular gene, a particular small part of that gene. And in both cases, the knowledge, the knowledge that we needed to identify that particular gene and that particular part in it was not produced, the knowledge was not produced by scanning procedures like GWAS or whatever. In both cases, the knowledge was the result of people who know about the virus, virologists, knowing what the virus is doing, what it needed to have available to do whatever it wanted to do, and then identify the part in the pig that you want to knock out to make that impossible for the virus. That is a completely new way of looking at things. We were always used at scanning genomes and not thinking about the deep biology of the process that we are trying to influence. And I think anything to do with what the GENE-SWitCH project and other projects that deal with annotation are trying to do it can only be that it will make it easier to do that kind of work again, because it gives you knowledge about how the biological process works, and that makes you much more powerful in developing technology to take control of the thing. Make sense?
it makes full sense, Peter. So in trying to summarize, mainly the outcomes of this project will increase the knowledge about the entire genome, and then the gene editing, like the example you made for the virus, will be facilitated by this increased knowledge.
I hope so. Yeah.
Yeah. Okay. Is there any other comment on this question? Everyone want to answer on this question? Please. Yeah, microphone. Yeah.
The knowledge generated in this project, of course, you need it to identify targets, because you have to be sure that you really have the functional variant that you are editing.
Yeah, exactly. This is what we were thinking. Yeah. Thanks for confirming. Any other comments or answer? Okay. Is there any question coming from the floor or even from the people on the table? Please, Chagla.
So I want to especially ask from the local perspective of the case of Italy or France or other countries in the Europe. What do you consider some regulatory or ethical considerations that should be taken into account when incorporating such research results like functional annotation techniques in pig and poultry sector? Or are there any kind of policy implications or other measures that should be taken to ensure the widespread use of these new technologies in breeding to reach to address some of the challenges that we are facing?
Anyone want to answer this Chagla question? Please, Paul.
I think we have sort of a antagonism here, because on one hand, it's all good and well for things to be free for everyone, but on the other hand, based on I don't want to get too philosophical, but
no, please be philosophical. It's interesting.
Sort of economic system. You really more and more the governments are not doing a lot of reducing or investment in research and so forth. So therefore relies on private companies to take up this information, and if they are not able to have a profit, then they just won't do it. And if there's not some limitation, then it's difficult for them to have a profit. So it's I don't have an answer to the question. I'm just pointing out.
No, this is an interesting comment. I appreciate it, Paul, because this is the hidden elephants in the room. Many times, the private companies have usually short and medium terms of to get return on investments, which the world, the humanity mainly should have a long term. But most of the research, good part of the research, is in the hands of the private companies, and people in this room will show it. We're able to show it. So this is really a problem. And I came back to you. I mean, thinking about this, again, the outcomes of this project, Paul, and as you work in FAO, which is so close to us in many aspects. And what do you think it could be the impact on the food security? The outcome of this project that is, as you say, is for high technological benefit and private companies.
Yeah, I think it's again, the main benefit is adding to the knowledge. It's maybe you can either look at upstream or very downstream relative to developing countries. You can say it's very downstream because if you look at Peter's figure, a lot of developing countries are not even at the level of 1950, and if maybe they don't even have the opportunity to use the phenotype of an own animal for selection, just because they need every animal that they need to use a male animal because it's available when the animal the female's in heat. So it's a big discrepancy. The other hand, you can say it's very upstream because it's a lot of basic research that needs more development before it can be used in the field.
Yeah, I agree. It's the breeding structure in those companies, in these countries, are completely different and really backward many years, and the lack of infrastructure will make difficult the implication. But as you say, is an increase on the develop of the technology and expertise. So sooner or later they will get them. Depends how late it will be. That's also important.
And I also will just point out that not so much my own unit, but we do a lot of work within the livestock field to try to encourage countries to improve the sustainability of livestock in general. And that's not just that's every country. So these results can be beneficial for those aspects.
Thank you, Paul. Please, come here. It's your house.
It's not my house. I had a question for Peter Knapp and also actually all of you. So since when I am in animal genetics, I hear about these very vague and wide phenotype descriptions like robustness, now resilience and so on. But I mean, from a biological point of view, can be a bit of everything. But of course, they make sense. I understand that completely. They make full sense for the industry because the black box still work. And maybe at some point, as you explained very well, there are limits, but it works. So the idea is, I mean, there is a lot of research ongoing to refine the phenotypes, to really have phenotypes that are more informative. At the animal level, you can have a recording system also for environmental parameters. I mean, so you can be very, very precise, having maybe something to really trace the single animal also for behavior and so on. And there are a lot of studies in this field. I wonder how much the industry is really moving on to adopt these things, because research is really moving fast. Maybe it's a matter of cost, I don't know. But how do you see this? How much are you going to invest in this phenotyping? Because phenotyping is fundamental to link, I mean, to understand how all these genome things make a difference. Thank you.
Some of our old colleague once 10 years ago say phenotype is king. I remember his slide and one presentation, EAP meeting. And yeah, Peter remember that.
Yeah, sure. And it is true. And it is a hot item. No, I think every serious breeding company is heavily investing in phenotyping right now. I have 3 colleagues in our team who work on phenotyping only in our genetic improvement team. And that is, yeah, that is all digital, yeah. A lot of usage of video cameras, etc. One of the main things there is analyzing the images. And yeah, that is a science all in itself. I've been playing with that for years and years without any kind of success myself. And suddenly, well, what you need is a couple of engineers who know how to deal with these things. The typical things here are, at least in our system, the typical focus is on behavior. For example, it's very, very difficult when you talk about things like tail biting in pigs or feather pecking in poultry. The main thing to do if you want to tackle it from a genetic point of view is to identify the actor, not the recipient of the behavior. And finding the actor is very, very difficult because you must see him do it. And what we are working on right now is extensive videoing of animals in their production environment with some kind of I don't know how it works, some kind of image analysis software. Just looks at the video and identify what the pigs are doing, and then spots a particular pig that is acting as a tail biter. And well, that way you can actually identify these pigs, and that is the beginning of a breeding program against that kind of undesirable behavior. And there are plenty of other examples, also in dairy cattle, and in beef cattle, in sheep. They work with drones that scan a huge part of the prairie in Canada, just to figure out which cows or bulls or whatever they are, what they're doing. It's very hot stuff.
it is very old stuff and phenotypes, systems, but in many aspects, or maybe in many countries, as the one we were mentioning before with Paul, for sure. But precision livestock farming is developing a lot for management, herd management, and I'm honestly, I'm not an expert of what is happening in poultry, but for the other species, I think they're developing a lot, and I believe
Yep.
the same in poultry. So I think the two things, there was maybe the hidden suggestion from Elisabetta, should be worked together. The improving the phenotyping through precision livestock farming developments and the genome research.
Yeah, it is a spin-off. Yeah.
Yeah,
The really important thing is the farming procedure itself. That is where let us say, where the money is going to be made. And the same thing happened, what was it, 50 years ago with milk recording in dairy cattle. Milk recording was there not because cattle breeders wanted to do it, but because the farmer had an interest in it. And then suddenly you discover, as a breeding sector, that you can actually use that data for the genetic purposes. And the same thing is going to happen here. The precision livestock farming is there for the farmer, but we can hijack it for genetic purposes because the data is there anyway.
But Peter, isn't it? Sorry, I have people who want to talk, but I just want to give an immediate answer to this. Is maybe the road was the opposite? No, when you talk about the dairy cattle, it started with a breeder organization who want to do genetic improvement. That's why they make phenotyping measurements, and then when there were no one supported, no national governments supporting this anymore, then the cost someone has to pay, and the farmers say, okay, we pay if there is some direct income to us, not directly genetic improvement. While in this case, as you say, is working in the opposite.
The opposite
but
side.
Both roads go to the same objective: using phenotyping for a better genetic knowledge of the animal.
Yep.
Sorry. Yeah, I tried to talk to the man. There was a lady before you. Sorry, the lady on the corner. Sorry, I forgot your name. I apologize.
My name is Barbara.
Barbara. Sorry, Barbara. Okay.
So phenotype is king, of course. We measure in the meantime nearly 100 different traits, but of course not on all our animals. And we are always searching for cheap proxies because you can't keep watching with video imaging thousands of animals. And to come to the added value of the GENE-SWitCH project, if you imagine one contribution could also be that you have a new phenotype measured on a relatively small amount of animals. If you get some ideas from a functional point of view where you might have to look for the genetic contribution, then of course once you manage to find a functional mutation that of course only partly explains the variants that you see, then you could also easier transfer that to other populations, or maybe develop from there also ideas for proxies that could help in the selection process. So that's where I would see the link here to GENE-SWitCH.
Yeah.
And more we see that, also the negative side effects of genomic selection, pleiotropic effects of these really large genes with large effects or variants with large effects, and also hitchhiking. And we really need this information to see and monitor better what we are doing in the genome. And then at some stage, you could also imagine that you are able to transfer that to third systems where you don't have this high technical impact on your breeding to find animals that add value in that process.
This is somewhere right. Paul will answer to you. I mean, I think I fully agree with everything you say. Just the last comment of what you say. Sometimes these developing countries, I believe they have different environment. So you cannot just take it from the Netherlands and bring into Alto Burkina Faso. So that is also a big constraint on this. This was a typical mistake happened not now, decades ago, when they took the best dairy cattle from Canada and brought into Africa. And a few months later, there will be parasites, everything that these animals will not produce anything. When it's like taking a Ferrari on a mountain track. I mean, a Jeep will go better there. So Please, Maria-José.
In fact, I also wanted to underline the fact that it's both a question of cost and of finding the right phenotypes that you are able to measure on lots of animals which are reared in groups, and you need individual phenotypes. So it's always difficult to find this kind of phenotypes, and annotation can help also finding the best proxies, as Barbara said.
Very good. Thank you, Maria-José. Well, I'm looking for other questions, and the discussion is getting very, interesting. And let's keep it going. Coffee can wait a little bit. Please, Chagla.
Yeah. Well, I want to ask. I want to ask this to Emanuela, Anna, and Stefania. How do you see these advancements? Like, okay, you had a very brief overview of what GENE-SWitCH is doing, or is going to, or is aiming at for the results. How do you think this would have an impact on the Italian sector? Because every country has its own environment, its own rules, or its own organization. How do you think it would affect the pig and poultry breeders here, but not limited to that, also the market and the whole sector?
I think it will take about a couple of hours to answer to this interesting question. But who want to start? Stefania, you want to start as a policymaker from the ministry?
Phone, okay, you can hear me. Okay. Okay, so for Italy, and improvement, genetic improvement of animal production is a main goal of the policy of the ministry, and this research offers tools to achieve this goal, and it's important also because we have tools that are very expensive. Most research can offer tools more available for all users of the sector, and it is important for the investment in these activities.
Thank you, Stefania. Anna, you want to say something?
Yeah, I can say something for poultry sector.
Yes,
of course, the genetic improvement, it's a key, because we have some problem with the broiler production, because broiler tend to grow a lot and have some problems with behavior. They have difficulties to walk, so for sure genetic improvement in some traits is very important to improve the welfare of these animals.
Okay. Before giving the word to Emanuela, you talk about welfare. What about the environmental impact?
From a genetic point of view?
Yes,
yes, of course. If we can improve the welfare, the animal feel better, and they maybe let's say are more efficient in the feed consumption, and so this can have let's say an effect on the environmental impact also.
Okay, thank you. Emanuela,
Okay,
from PIC Italy?
Yes, from my point of view, I think that functional genomics is very important to speed up the genetic improvement at the farm level. However, we need lots of investment, and of course, we need to have gene sequencing in most of our population. So I know there is a project for the ministry that give the opportunity to have the genome sequence of a sample of different species population. So we could really get lots of data back from this sequencing, and of course find which are the main genes that can impact the traits that are not easy to measure, like, as we say, reproduction traits, also behavior traits.
Thank you very much. Elisabetta, please. Maybe you had to come for the microphone, Elisabetta, sorry.
Manuela parlato, so you mentioned a project that is not a PIC project, it's a
No, it's
Italian
government.
So PNRR.
Yes.
Yes, thing. So, I mean, what is the plan or the idea is to sequence a lot of populations?
Yes, to have the genome of
And then
different
to make
species that
imputation
are
then.
of economical impact in animal
So
production.
characterizing really the diversity, also the different breeds and so on.
Yes, we can collect lots of data in Italian database and work start from that.
Okay. Do you know if it will start this project?
I know in Dairy, yes, it started a
Dairy,
few
okay.
years ago.
Okay, but now with new funding, because you know Italy is getting in principle a lot of funding
Money,
from Europe, and so a lot of it should be used for new research projects, correct? So
yes,
I mean, so this will continue. It will be used, this money, also to expand on this. That's good to know. Thank you.
Okay. Thank you, Elisabetta. Thanks. Thanks very much. Comments, questions? If not, then I'll go back to the point, well, some ideas coming from the answer that we got now. I mean, talking what Emanuela Parlato and Anna said, actually there is this policymakers' willingness all over the world to take care of sustainability. This is a global keyword since few years, and I think is right. And might not be the same objective of the private companies. And because of course private companies are looking more for direct income, which is obvious, also logic is in their logic. So how these two different logics work together? How can the policymakers support the development of the industry, or the poultry and pig industries, and how the poultry and pig industries see the point of aiming at sustainability and putting this in their breeding objectives? It sounds like quite a kind of vague question, which it is, but I'm expecting you have some. I got some views from maybe from Anna, who was silent so far, and she represents the European animal breeders. What do you think, Anna? Did I catch you in the wrong moment? Huh?
We can't hear you.
Anna, we love to hear you, but we can't.
Can you hear me? Oh, yes, right.
Yes,
I'm green now. Yeah, it's a very broad question, so I don't know. From what I can say first, what is sustainability first? That will be because that can be many different things. It will depend. Somebody said different countries, different regions, I would say. Italy in the north is not the same than Italy in the middle or in the south, or even close to the sea or in the mountains. So what is sustainable in one place is not sustainable in other places. And of course, this to answer in a general way, as we were asking Andrea, we have the code everywhere. So breeders are already responding to resilience and sustainable sustainability challenge. So, and that's why we have drafted this code of good practices. But again, I think that the point of policymakers is, are they understanding what breeders are doing? That's my main worry at this moment, is because when we talk about sustainability, if you take the environmental pillar of sustainability for poultry, for example, it is better, and that's the problem of our policymakers, is that they are saying this is good and this is not good, and they are not considering in between all the possibilities, or not always. But in poultry, let's say that organic has some benefits, And conventional has also benefits. And from an environmental point of view, or the environmental pillar of sustainability, perhaps the organic is not the best, but for other pillars, this is probably good. So my point is how policymakers, they assess sustainability depending on the systems, the regions, and the farmers that are working in. And that's the complexity that we have to raise at this policy level. So this is not only good, or this is not only bad, but yeah, and that's what we are doing with the Code FAVAR, to try to answer to this and to show all the differences between breeding programs and companies and associations working in the field. I don't know if I'm responding to your question, but it was so broad.
Well, you answered in a good percentage of my question, but at least you open new roads. And I'm using one of these roads to go to Peter, because what Anna says is that sustainability is not the same all over the world. It's not the same in a country like Italy. It's not the same in Europe. It's not the same in the world. But your company, the company you work with, for Peter, is a global company. PIC works all over the world. And do you have a different kind of approach in genetic, in functional notation of genome and pig genomes for sustainability? I mean, first, if you have it, and second, if you have it, did you differentiate considering where you apply this research?
No, the differentiation across the world that we talk about has much more to do with breeding goals, in which traits do we focus on, than with the technology behind the breeding value estimation. We estimate breeding values in a particular way at this moment with genomic blood, without annotation, etc. That is likely to change the coming 5 years. But we don't do that in a different way in Asia than in Africa also. The differences have to do with which trait is important. In this region, in another region, there may be another trait of importance. That leads to different breeding goals, different selection indexes, if you want. But coming back to the other half of your question, Andrea, how can policymakers influence things that the breeding industry is doing? I think that's very simple. We must always bear in mind that the animal breeding industry does not have its own agenda. The animal breeding industry must produce animals that somebody want to buy, want to pay for. And if somebody wants to pay for a particular animal, for a particular type of animal, then some breeder will make it. Which means that if policymakers or whoever influence farmers, producers, for example, by making it more expensive for a farmer to produce a lot of carbon dioxide, and that is not very difficult to do, then the farmer will, then what we will have is a demand for animals that produce a lower amount of carbon dioxide. And then the breeding industry will make that animal. That is how it works. It is a supply and demand thing, and it happens to be so that we supply, but we do need the demand. If there are no farmers in the world who want to pay extra money for an extra green pig, then the extra green pigs will not be developed.
No, that is clear, Peter. I mean, your company, like any other private company, works on market demands. Market demands that you produce.
Not only private companies. Exactly the same thing holds for every kind of herd book organization.
Yes, yes. But the policymakers should be, in theory, a step ahead and planning the future world for the humanity, which, of course, with all the type of constraint. But you say, if the policymakers say want to have a green pig, and the farmers in Brittany, in Po Valley, or wherever in the world must have a green pig, you will give a green pig to the market.
Yep.
I mean, that's it.
Yeah, Andrea, but what is a green pig? I mean, that's my point. We go back to the poultry, the organic poultry and the conventional free range and so on. They have all benefits for the environment, all. I mean, organic is good, is better for biodiversity. I'm simplifying, of course, but they are using a lot of land and they are using more feed than conventional. But at the same time, conventional, Anna was the other Anna with two N's. She was she was saying that there are some issues on welfare. So what the Commission, I mean, the European Commission is proposing at this moment is that we are going to discard the lines that are growing faster in Europe, because for them that's not welfare. But from a green perspective, could be a bad idea. But I mean, that's what I wanted to say at the beginning, is that what is sustainable is not a definition that is the same for everyone, because it has many different aspects in. And that has to be understood by policymakers. And I'm not sure, I mean, probably they do, but they have other people pushing in other directions. So organic is not the final green pig, poultry, or whatever. It will be good in some environments, in some places, but it's not the final, should not be the final objective of policymakers. And we have the feeling that it is. And we can open the scope and talk about other things, not only breeding, I mean, not only welfare, but that's the main, and animal welfare legislation is not aiming to take part on this discussion of sustainability, or at least they analyze the assessment they are doing of the impact of this draft new legislation is not giving to sustainability the weight they should. So that's my problem with policymakers, but perhaps it's because I might be tired of this animal welfare legislation discussion.
I think, Anna, you're doing a incredible job in Brussels. Try to convince those people, and I believe that, as you say, they are convinced. The problem is there are voters that I think about something populistic ideas, and I say in the worst meaning of the word populistic ideas to follow. And as you say, okay, organic, but then we have a problem with CO2 and global climate change. But that's why it's important researchers are close to policymakers as much as possible. I know it's a very difficult job that you are doing, Anna, and other people are doing for other sectors. So, but that's what we have to do. But I think these topics are incredibly interesting, but we are diverging a little bit from the GENE-SWitCH point. So I will go back to this and talking about, to the policymakers here around again about the application of the functional annotation on pig and poultry genomes. How do you support and is the legislation working on this part? Because at the end, as Emanuela Parlato said before, there is the Ministry of Agriculture in Italy and other ministries around Europe and the Commission. They are working in the same direction, and overall they supported these projects like GENE-SWitCH. So they should get an outcome and utilize this outcome. Otherwise, it can be, I don't want to say lost, but not used completely. So what is the policymakers thinking about the potential outcomes of GENE-SWitCH and functional annotation? I don't know whom can answer this. I think the policymakers, I can see maybe Stefania as maybe and maybe representing breeders organization, Anna, or FAO with Paul. You want to give some comments on this? Who starts? Don't be shy. Stefania, are you there? She's muted. So Paul, I'll ask you to say something, please.
Yeah, well, I think the policymakers will maybe just again in the general sense that
very general,
adding to the knowledge base, looking at that these kind of results can improve sustainability, or one aspect in sustainability, whether it's more efficient production or some way to maybe these approaches for annotation can be used in some way to measure, improve the welfare and so forth. So I think that's
the way.
Yeah, just have an idea that this research eventually will contribute to the knowledge and improve society. So therefore, you need to continue research, even if the company's not going to take it today and use it tomorrow, sort of thing.
That was the hint I was going to look for, to continue to do something. But we go back later. And Anna, be prepared because I'm going to ask it to you. Anna, can you want to answer to the question I said before?
Yes, I think that the policymaker can help the application of the genetic development and improvement also by helping the breeder company or the farmers in the real application of this new knowledge, because as we said before, it's a matter of cost also. So probably this can be a big help for the application in the field of this new development.
Okay, thank you, Anna. Anna, based on what I asked before and the little hint given also from Paul, do you know, since you are in the important rooms over there in Brussels, if there is any potential follow-up of this project like this one, and in the future or in the horizon that is already in the calls that are already out now? This is important for our scientists here and the stakeholders.
I think, well, there are two things in your question. I mean, at least for me, it is how this project can, the results of this project can be taken to for further research, right? For further calls of the Commission, and the second one is how the knowledge of this project, but not only this one, because we have also BobRec and what is the third one, guys? Aquafang. Yes, thank you. What we can not influence, but can deliver a translation to policymakers to let them know what is the potential of the research to improve animal breeding or and so the animal farming. So for one, there is for the first question, sub-question that I have identified in your question, Andrea, is, well, we are building already a Eurofunk research infrastructure. I would say that's part of the thing, and perhaps what we have to do is we had already 2 weeks ago, but this was a very narrow conversation because I was about two very specific topics at the annual meeting of Faber TP. Perhaps what we have to do is to make this exercise with the industry, with the private sector, with Aquafunk and BobRec, and to deliver a common policy brief to the Commission for the the strategic plan of Horizon Europe 25-27 in the framework of Faber TP, if you want, because we are already doing the same. That's part of a question, a response for your question. But the second one, I think we have to translate better, and that's our job. If and you, the team, Andrea of EAP, in projects, is that we have perhaps to better communicate to policymakers the outputs of the projects, and we have to really translate to policymakers that they don't know what is epigenetics or functional annotation or whatever, or even precision livestock farming.
Yeah.
So
and this is a much more difficult job.
Exactly, and that's a job that I will say.
Job.
Yeah, but that's a job that I think we can do.
Oh, we will.
And we have to do. But the scientists also have to trust us to do it.
Yeah,
I would say, and let us forget all this scientific wording that a lot of people, they don't understand. And I think most of them, they do. But it's very difficult to summarize 4 or 5 years projects in one page. But we should do it. I mean, that's a key point. Yeah.
Thank you, Anna. Thank you very much. I share everything you say as usual. And there are any other comments from the audience, from the speakers? Please, Barbara.
I have a tiny example. We use the annotation information to find deleterious variants. And if we avoid losses, not also before birth, but there are also things that happen before weaning or during the process of growing, then of course we reduce the environmental load and the losses, we reduce mortality. So that's a little example where we can use this information, also increasing health.
Yeah, all these acquired knowledge, yes. Yeah.
And improves welfare, and it reduces CO2 footprint. So
exactly.
These would be nice examples to add.
Thank you, Barbara. Elisabetta, please come on the mic.
I think that a big merit, not only of GENE-SWitCH, but of the fungus in general, has been that we have made a lot of effort, including in this project, on standardization of data, metadata processes. And all this is open share. It's open. And I think this has been probably the biggest merit of a FANG initiative, because this allows really a lot of development, full access. And also for policies, I think it allows that, I mean, people will see our FANG movie, our Eurofang movie, and we'll go to see, we'll be curious, and we'll go to see and access the data and maybe have ideas. So I wanted just to say this because I think in this we are quite great.
Good, I'm sure we are great. And standardized, I think, is the most complicated job to do. In a situation like this, when you talk about biological data as they are, standardizing is a hell of a job. Also render public is things. And it goes back to something what the issue with polls, you know, going also to these developing countries, because everyone can use it if they have the expertise to use it. Okay, if there are no other comments or questions, I want to thank everyone in the audience here, and Peter, Anna, and Stefania were online. Peter and Anna wait for you in Lyon. Don't miss it. I will look for you.
No, we cannot. We are a bit obliged.
Not going to miss it at all.
But the rest of the team too, Chala and Gina will be there with Nora, so no problem.
Okay, thanks to
and
Chala.
I think Peter has 2 presentations to do, I think, or 3. 4. So
it means if we get some problem before Lyon, we don't have the meeting in Lyon because Peter is not coming. So, okay, thank you very much. And thanks to Chagla, Emanuela, Paul, Anna, and Marie-José, and Barbara. Thanks a lot. Bye-bye. Thank you.